Thank you all for coming. I'd just like to start by making the acknowledgement of country. I celebrate and acknowledge the traditional owners and custodians of the land on which we meet today, the Wurundjeri people of the Kulin Nation. I pay my respects to their elders past and present, and I embrace their continued connections to this place. It's my pleasure to welcome Tim Stinear, who's probably well known to most of you because he's in the Parkville precinct. He's from the Doherty Institute, and I invited him over because I still read The Age on weekends, and I like a bit of soft science, despite being a scientist. Not that I'm saying that your content of your work is soft science, but if you were like me about 6 months ago, you will have seen an article, I think it was in the Good Weekend in The Age, and it talked about the spread of the Buruli ulcer, also known as the Bairnsdale ulcer or Gippsland ulcer. It's got a lot of different names, and how it's now coming, this kind of third world disease is now kind of invading the peninsulas we know and love, and many of us kind of go on holidays to. So I thought, given our interests in population genomics, in fire and so on, and our interests as an institute here locally in terms of diseases such as this, I think it would be a good idea to hear about this from one of the world leaders in this group. So just a little bit more about Tim. He's the head of genomics over at the Doherty, and he's situated in the Department of Microbiology and Immunology. He's an NHMRC Senior Research Fellow. And he did his PhD at Monash in 2001, and then had a stint at the Pasteur, and then came back and has been working at the Doherty since then. And as I already said, he's a world authority in the genomics and genetics and molecular biology of Mycobacterium ulcerans, the Buruli ulcer. So I'm sure you'll find this talk interesting. Just another note, I haven't organised any meetings with Tim. I figure because he's a local, I'll leave it up to you if you'd like to chat with Tim, and I've alerted him to the fact he might get people running down his door after this seminar. So welcome, Tim, and we really look forward to hearing what you have to say. Thank you.
Thanks very much, Melanie. All right, soft science, here we go. Actually, it will be a bit soft. There's some sort of hard bits sprinkled in it, but the brief from Melanie was to give an overview of the emergence of Mycobacterium ulcerans disease on our back door. So I've presenting some of our own research, but it's an overview where I've intermingled what we know about Buruli ulcer and the causative agent Mycobacterium ulcerans, and why we think it might be spreading locally. And I've been trying to think of good ways to communicate what's going on with this disease. Sounds like thunder up here. Is that normal? Is that? It'll go away, will it? Right, okay. All right. So I've come up with this. It's a little bit cute, but let's just go through the story of a local Buruli ulcer patient from 2017. So his name's Luke. He's a 30-year-old male. He's married. Why that's important will become evident later. He lives in Coburg. And in July 7, 2017, he noticed on his elbow just a little papule, like a mosquito bite that wouldn't heal, slightly raised, red welt. Wasn't painful, so he duly ignored it. But so that was July. In September, he goes to his local GP to get it looked at because it's much more angry now. It started to break down a little bit. Still not painful, but it's certainly not healing, and he's a little worried about it. The GP said, it's probably a staph infection, and gives him a third-generation cephalosporin or something like that to treat it as a staph infection. So October 2017, the lesion has now progressed in size, a little bit painful, but not obtrusively so. Just keep the dressings on it, keep taking the antibiotics, but it's still not healing. So Luke's worried. He goes back to the GP. They continue the staph treatment, maybe change up the antibiotics a little bit. Now, I should put a warning with this. This is what Luke's elbow looked like by November 2017. The ulcer was extensive, extremely painful now, and he consults Google. And Dr. Google says to him, amongst many things, leishmaniasis, all sorts of things, but it could also be Buruli ulcer. So he does a bit more Googling and suggests to his doctor, even though he lives in Coburg and it's not a known Buruli ulcer spot, he says, maybe this is Buruli ulcer. And the GP says, what's Buruli ulcer? And so Luke explains what it is. It's an infection caused by a bacterium. It's known in Melbourne. No, it's not. It's a tropical disease, says the doctor. But anyway, there's a bit of to and fro, and eventually the GP takes a swab of that lesion and sends it to the Mycobacterium reference lab here in Melbourne at Vidril, which is just in the Doherty Institute. They do a very sensitive and specific PCR, and they diagnose the presence of Mycobacterium ulcerans on that swab. And so this is Buruli ulcer. Luke has Buruli ulcer. So a little bit more about about Luke's condition here. This is another elbow infected with Mycobacterium ulcerans of a patient of Paul Johnson's from a few years ago. And the important points to note is you have this central zone of necrotic tissue with this undermined edge here around this central zone of necrosis. Now, if we were to take a section or a swab of material from this necrotic centre, in fact, we could take a probe and push through this necrotic tissue and into necrotic subcutaneous fat beneath this relatively intact dermal layer here. And we would see, if we do an acid-fast stain for Mycobacteria, pools and pools of acid-fast bacteria growing extracellularly in these zones of necrosis. And what we'd also notice if we changed our staining is a marked absence of inflammatory cells. These are mycobacteria, and mycobacteria have a cell wall made of mycolic acids. These are potent adjuvants. Freud's adjuvant is essentially a mycobacterial extract. But yet here we have a lot of antigenic load, highly antigenic bacteria, and you can see there is inflammation here, but in proportion to the amount of bacteria, surprisingly little. And also remember, Luke didn't report really any pain associated with his lesion until it was quite developed. So there's a painlessness and an absence of inflammation, which are hallmarks of this condition. Now, there are several different presentations, in fact, of Buruli ulcer. You can have an edematous form where you get extensive swelling, still with the subcutaneous necrosis, the bacteria replicating in your subcutaneous fat. You can have an osteomyelitic form, an infection of the bone. But Luke's not in mortal peril. He's not going to die from this condition, and I'll talk about the treatment later on. But it will leave him with considerable morbidity and lifelong deformity that he'll have to learn to live with. Okay, so what is Mycobacterium ulcerans? Well, a few years ago we sequenced the genome of Mycobacterium ulcerans and established that it's actually closely related to Mycobacterium tuberculosis, the causative agent of human TB. But it's different, it's special. M. ulcerans has the genomic signature of a niche-adapted pathogen. What's another example of a niche-adapted pathogen? Mycobacterium leprae, cause of leprosy. Or Yersinia pestis, the cause of human plague. These are bacteria that have downsized their genome, undergone metabolic streamlining to become good at doing one thing only, living in a particular niche, living in the midgut of the flea or whatever it might be. And we see the same signature. This isn't the genome of Mycobacterium ulcerans. This is just a plasmid that it has, a special plasmid. But its genome has undergone reductive evolution. And the bacterium, as I say, looks like it's adapted to a niche environment. And you're sitting there, well, what is this niche environment? And I really don't have the answer for you. And it's something that's eluded us to this day. But it's not a generalist. I'm going to talk about, the environmental burden of Mycobacterium ulcerans, but I really don't want the audience to go away thinking that this bacterium is probably spread everywhere in the environment. It's niche adapted. It's living in a protected environment where the things that, a bacterium needs to live and survive are being provided for it fairly readily and doesn't have to scavenge as much. But one thing it's done over its course of its evolution is acquired this plasmid. And this plasmid has 3 very large genes that are about 50 kilobase pairs here, these two, and a smaller 7 kb here. These encode polyketide synthases. These are molecular assembly lines to produce polyketides. And this is the polyketide produced from these 3 enzymes encoded here. It's called mycolactone. It's a macrocyclic polyketide. It belongs to the same class of compounds as the antibiotic erythromycin. Sounds like my lunch, but it's, but I assure you it's not. Anyway, I don't know what's going to happen next, but thank you, Weehai. It's probably a soft science indicator. Once I hit a certain threshold, it'll go away. All right, it's my own fault. Anyway, this is mycolactone, 12-membered ring, macrolactone ring with these 2 acyl side chains. And it's produced, as I said, by this polyketide synthases encoded on this rather extraordinary plasmid. Now, Pam Small was the Rocky Mountain Labs investigator that discovered mycolactone in the late 1990s. And since then, many people have worked on it. And I'm just going to summarise lots of work in 1 or 2 slides to say that we think, well, mycolactone is the primary explanation for the pathogenesis of Mycobacterium ulcerans. All right. What's it likely to be? Does anyone know? Is it?
I was wondering if there's something running with construction.
I don't think it's the audio, is it? I've left my phone down there. There's no.
It's it. It sounds like the whole floor is running.
I could just push on. Oh, well, we'll just push on.
Yeah.
But yeah, no, it's okay. It's okay. Look, just a summary of what we know. Actually, this is the summary. Our best evidence to date is that mycolactone specifically inhibits the alpha subunit of the Sec61 translocon. And I'm sure there's people in the audience that know more or perhaps even work on the Sec61 translocon. But data to date suggests that this molecule specifically inhibits the transport of proteins across the ER membrane, so specific secretory proteins. So, for example, it impacts cytokines and plasma membrane receptors. And one of the consequences of this disruption is that the host is unable to mount effective immune responses. Prolonged exposure of different cells to mycolactone leads to apoptosis and cell death. And I've taken this summary from Caroline Demongel's recent review that sets out some of the features of the pathogenesis or the pathology of Mycobacterium ulcerans infection and how we think this is explained by this Sec61 blockade. So skin ulceration, you can see impact on keratinocytes and endothelial cells, depletion of thrombomodulin to explain coagulative necrosis. If we look down here, local analgesia, this absence of pain, at least in the early stages of infection, it might be explained by the impact of mycolactone on pre-apoptotic neurons, Schwann cells and microglia, altered cytokine production by neurons and Schwann cells, and inhibition of inflammatory pain has been demonstrated in different rodent models. And then we have these deficits in local immune responses and systemic immune responses that we think are partially or fully explained by the impact of mycolactone on Sec61 in different cell types and in different contexts. Anyway, there's much, we could give a whole seminar on mycolactone. But anyway, M. ulcerans, it's a special mycobacterium. It's developed this capability to produce this small molecule, mycolactone, that has a range of profound effects on mammalian cells. But back to Luke. So the story of Luke. So here he is. He's now ready for treatment. So it's from December 2017. He now starts a combination of rifampicin and clarithromycin for 8 weeks. Now these are fairly high-powered, high-strength antibiotics that are not without side effects, but they do cure this infection. He has some paradoxical response because once we start the antibiotic treatment, the infection is effectively unmasked, and so there's a strong immune response now mounted to the huge antigenic burden. So he would augment his antibiotic treatment, perhaps with adjunctive corrective surgery to debride and reduce the burden of the infection, and also perhaps some cosmetic surgery as well to improve the healing response. Anyway, September 2018 comes round, almost a year after his initial diagnosis, or after a year, and he regains almost full use of his arm. He's left with contractual deformities, he's got a large depressive scar, he has a lifetime of disability with his limb. And Luke's going, well, where did I get this infection from? How did I get Buruli ulcer? I just live a nice, quiet life in Coburg, minding my own business. How did I get such a terrible condition? And I want to take a step back and go to, well, what are the origins of Mycobacterium ulcerans disease in our region? And actually the story, at least in modern times, say at least from the 1930s onwards, begins in Swifts Creek, which is in the East Gippsland area near Bairnsdale, where people from farming community, children and their farmers from small community around Swifts Creek in the Bairnsdale region, presented to their local GPs with these unusual skin ulcers. And the initial diagnoses were, this is cutaneous tuberculosis, or this is leprosy, because they saw the acid-fast bacteria in the tissue specimens. But none of these diagnoses were sustained. And it took a team of distinguished scientists, including our own here, Sir Peter McCallum, but also 2 hospital scientists, Glenn Buckle and Jean Tolhurst, Harold Sissons here, who published this seminal paper in 1948, fulfilled Koch's postulates and showed that the infections in these people from around the Bairnsdale area was caused by a new, previously unrecognised mycobacterium called Mycobacterium ulcerans. Since then, it's become obvious that the disease was already existing in Africa, and there have been many cases reported, although they're not correctly diagnosed, from Africa and in other places around the world. So now the WHO recognises Mycobacterium ulcerans infection, and its official title is Buruli ulcer. Buruli was a county in Uganda in the 1950s where there were many hundreds of cases reported. But it's a neglected tropical disease, so it's been reported in, well, between 2002, 17 countries, but more than 33 countries worldwide have reported Buruli ulcer at one time or another, mostly in West and Central Africa. It's a disease of poverty, and it's a disease of the rural poor of West and Central Africa. 55,000 cases counted between this time period, over 17 countries. Where are those countries? As I said, mostly West and Central Africa. The depth of the green is the indication of the number of cases reported. And look, this isn't incidence, okay? This is a neglected disease, so we have a very poor handle on what is the true incidence of this disease globally. So these are cases that are reported by these countries. And you'll see Australia is a massive green mass. Now, as you'll see in a minute, the disease is not present everywhere in Australia. We have a focus here in southeast Australia, in particular around Melbourne, but we have sporadic cases up the eastern seaboard, virtually nothing between Eden and then north of Brisbane, and then a few spots here in Queensland, and then another focus in the Daintree region of far north Queensland, and then a few other cases reported from around Darwin. But it's relatively rare in Australia, or at least it was, with most of the disease occurring here in southeast Australia. Just to give you an example of what's going on, say, in West Africa, here's an example of the disease from a young woman in Ghana. Again, an elbow. It's not always the elbow, as again, you'll see later on. But here you can see the probe here pushing underneath relatively intact skin into this necrotic tissue. Where does the disease occur? It occurs around slow-flowing water, swamps. It's associated with wetland areas. Now, what is the environmental reservoir in West and Central Africa? We don't know. We've looked extensively, but the association between these sorts of environments and the occurrence of the disease in humans is very strong, okay? But we do not have a good handle on the environmental reservoir. There is a role for aquatic insects, but that's not something I'm going to dwell on too much today in the African context. Okay, but these are the questions that have troubled the Mycobacterium ulcerans community since the 1930s. And a very famous and distinguished Mycobacterium ulcerans researcher, who I won't name but is actually in the audience, did say to me once that I would really like to know what is the reservoir of M. ulcerans and how is it transmitted before I shuffle off. Now, we're getting closer to helping him answer that question, but as you'll see, we're not quite there. There's a little bit more to do. So, John, we haven't given up yet. Oh, did I say that? All right. Okay, what about the local situation? What's going on here? So this is the traditional endemic area for Buruli ulcer in southeast Australia, and it's the Gippsland area here, around Bairnsdale, as far north as Mallacoota here, even into Eden there's been a case, and around the Gippsland Lakes area. But in the 1980s, something happened. Cases were reported here at the top of Western Port Bay, around Warneet and Tooradin. And in the early 1990s, there was a very infamous outbreak of the disease involving about 35 cases at Phillip Island. We know this thanks to the efforts of a particular Melbourne pathologist, Dr. John Heyman, who actually interviewed all patients virtually between 1965 possibly up into the mid-2000s. And so we have very good understanding of the local epidemiology thanks to the efforts of one individual. So disease bubbling along here in the 1930s, perhaps earlier, and then jumps in the 1980s. We start seeing cases here around the top of Western Port Bay, outbreak here in the 1990s, and then sporadic cases around here. And then in the 2000s, we start to see cases on the Bellarine Peninsula at St Leonard's, and then a little bit later around at Point Lonsdale. And if we start looking at now at incidence data, because in 2004, the Department of Health said, right, we want this disease to be notifiable. So we now start to get data gathered by the health department, not just relying on one individual doing this in his spare time. And you can see that the cases here, possibly hard to read the axis there, but this is the incidence. I've chopped it off actually. This is total number of cases here, and you'll see a minute the incidence value on the other y-axis, which is fairly low. But cases nonetheless starting to bubble along in the 2000s. But then this has happened. And this is why, Melanie read about it in the Australian magazine a month or two ago, is that we've seen this tremendous increase in cases, and now the incidence is 4 per 100,000, and this is the total number as of this week is about to surpass the total number for 2017. So we're looking for a record year of cases of Buruli ulcer coming from Victoria. So something's changed. And if you want to see the spatial representation of those data, or at least portion of them, I'm just going to show you a series of maps that have not worked in reverse order. Right, no, that's right. Okay, so here's the time at the top and the number of cases. And if you see here, this is Barwon Heads and Point Lonsdale, or Ocean Grove and Point Lonsdale. And you can see the heat map signatures here of cases. But we're also seeing a little sprinkling of cases around the bay here, but not very many, a couple at Sorrento. If we jump forward to 2013, 2014, we now see the disease has taken hold on the Mornington Peninsula and spread along from Sorrento through to Blairgowrie and into Rye. And this is data from Michael Loftus and Paul Johnson and colleagues, published, soon to be published. And if you look up to the end of 2016, you can see that it's sort of dying out here to a certain extent on the Bellarine Peninsula, but really going gangbusters on the Mornington Peninsula. And not just on the Mornington Peninsula, you're now seeing Frankston, Seaford, Beaumaris, Hampton, also starting to report cases. So, Luke's starting to get a bit concerned, I guess. Where or how did I get this infection? Well, Luke, did you go to any of these places? And you have to think about that, Luke. We'll come back to that in a minute. Because there's one more thing to tell you about reservoir. Remember I said in Africa we don't know what the reservoir is? Well, that's not quite true here in Victoria. So we've been looking for some years, a team of us have been looking for some years to try and find a reservoir of Mycobacterium ulcerans in the environment. Because the epidemiology, actually I should have told you this earlier, person-to-person spread is extremely rare. There's very poor evidence for person-to-person spread of this infection. But we discovered, a team of us led by Janet Fife at Vidril and Paul Johnson and colleagues, that the Australian native possum, the common ringtail possum and the common brush-tailed possum are a reservoir species for Mycobacterium ulcerans. They're a susceptible host. They develop Buruli ulcers. This is a tail with a Buruli ulcer. And they shed the bacteria in their fecal material. You can have up to 10 to the 6, 10 to the 7 bacteria per gram of possum fecal material. And so we can imagine now a very large environmental burden of the bacteria being shed into the environment, thanks to our possums, which, as we will all know if you live in Melbourne, are ubiquitous and live in and amongst us very happily. And if we go and survey, now the possum fecal pellet is a very handy analyte to go and look for the presence of the bacteria in the environment. We can just go and pick up possum fecal material and screen it by our molecular test for Mycobacterium ulcerans and get these sort of maps then. If we just sample every 200 in these quadrants of 200 metres, we stop and look for possum fecal material, we pick it up and we log its position and then we test it. And the red spot here means it's M. ulcerans positive. And what you can see, this is a map of the Blairgowrie-Sorrento area. And again, this is work from Janet Fife and colleagues, is that, see all these red spots? These are possums that are shedding M. ulcerans positive fecal material. And this circle here shows us that within this circle, there were 4 residents and 2 visitors to that area that developed Buruli ulcer. And if you look in the rectangle here, there was an additional 2 and 1 people. So there's a nice correlation here that, between the presence of possums carrying the bacteria and the likelihood that a human entering this area will develop Buruli ulcer. So we can sort of explain all this to Luke and say, oh, well, it might be the possums, but I don't touch possums. I hate possums. I keep clear of them. And I don't remember going to any of those places. Well, actually, Luke did remember going to one of these places. When the health department asked him, where had you been? Have you been to any of these sites? He said, well, on Mother's Day, I went to visit my mother-in-law for an afternoon in Rye. And so he'd had about a 4-hour exposure to a known endemic area. And there have been several cases like this of these very point exposure examples. And again, Paul Johnson has collected these with help of registrars and the Department of Health Epidemiology team. Actually, the point exposure people give you a very useful way to establish the incubation period for the disease. So from the time you enter an endemic area to the first onset of symptoms. And we now know from a look back of more than 600 cases that that incubation period is 4 to 5 months. So very slow. But in addition to the short exposure periods, Paul Johnson, infectious disease physician at the Austin Hospital, deputy director of ID there, longtime Buruli colleague and researcher, noticed these sorts of cases. This is a young girl with a Buruli ulcer lesion on the top of her ear. And this made him think about mosquitoes. Maybe biting insects, or mosquitoes in particular, because they're so prevalent, are having some sort of role in the spread of this disease. And so he initiated a large survey back in the mid-2000s around the Point Lonsdale area where we trapped tens of thousands of mosquitoes and screened them for the presence of the bacteria. Maybe this table's a little hard to read, but essentially it's this column here, the maximum likelihood estimates, the number of mosquitoes carrying M. ulcerans was 4 per 1,000 here in the Point Lonsdale area. And the further away you get from an endemic area, so if you end up in Mildura, you have no mosquitoes positive for M. ulcerans. So there did seem to be here, bacteria, mosquitoes in area where the disease is present, the mosquitoes are also showing signs of the bacteria. Paul Johnson did a few other things looking at associations. This plot here is showing you different disease profiles. And if you take an arboviral, or if you take a disease like legionellosis or tuberculosis, this is their incidence profile over time. If you look at an arboviral disease like Barmah forest virus or Ross River virus, they have this sort of seasonal pattern here with mosquito, in line with mosquito population changes. And if you just change the axis and look at the incidence of Buruli ulcer, it mimics the arboviral diseases. He also extended that association between place and whether you were mosquitoes carried M. ulcerans or not. And this is Point Lonsdale. The rate of disease in humans mimicked the rate of carriage of mosquitoes with M. ulcerans. And the further away you went from the endemic area, the less likely you were to see human cases and positive mosquitoes. Now, Paul, while I'm presenting his data, did one more study where he got a medical student to precisely map on a human body where lesions are occurring. And there's a very non-random pattern of disease occurrence on the human body. This is the number of cases, this is the front of the body, and you can see the lower legs here, and this is the back, and you can see the calves and the elbows are common. Now, does this prove nothing? No, it doesn't prove mosquitoes are involved. But it certainly doesn't discount the possibility that they might be involved. Okay, these are certainly potentially exposed areas of the body. All right. One thing we wanted to help Paul with, or I wanted to help Paul with, was, we're inching here towards establishing an agent, a microbiological agent vectoring a disease. And there's very well-established criteria in the field for showing this, trying to show this. This is the Barnett criteria, one set of criteria that are used to show whether a particular microorganism might be vectoring, an insect might be vectoring a particular infectious disease. And here they are. The vector must be shown to acquire the pathogen from an identified source. Vector has close association with infected hosts. Vector must be found infected with the pathogen. And then this item 4, efficient transmission to competent vertebrate hosts must be demonstrated experimentally. So this is where we tried to help Paul because we had done much of this association data in these first three, addressing these first three criteria. So we tried to address this specific hypothesis first. Imagine that you've got skin that's contaminated with Mycobacterium ulcerans. You've been working in the garden, you're up to your elbows in possum fecal material because you've been digging in your compost. And what would happen if you got a puncturing injury, either via an insect or perhaps a needle stick injury from a prickly bush, something like that? So we set up the following model. Start simple. We took anaesthetised bulb C mice and we coated their tails in a culture of Mycobacterium ulcerans. We developed a bioluminescent strain of Mycobacterium ulcerans so that we could follow the progression of the bacteria more readily using IVUS, and also just to show here that we did have some sort of relatively homogeneous coating of the tails of the animals. We then exposed these anaesthetised mice to cages of about 20 mosquitoes, and we let the mosquitoes blood feed for a defined period. And in this particular set of experiments, we used a local mosquito that's common on the Mornington Peninsula, Aedes nodoscriptus, and we also used an exotic species, Aedes aegypti. Why did we use this? Well, this was hard to get hold of, and then we had lots of this thanks to our collaborator Ari Hoffman and Jason Axford at Bio21. Okay, so numbers of mice in each experiment and the number of mosquitoes per mouse, and then we also observed the number of mice that were bitten during these experiments. And I'm not sure how well this projects, but hopefully you can see a couple of mosquitoes that are sitting on the tail of a mouse up there, protruding in through the cage. What did we find? With our Aedes nodoscriptus experiment, you can see here that 2 of the mice that were bitten by Aedes nodoscriptus went on to develop Buruli ulcer. This is the PCR result, the histology scores, and then the CFU of bacteria per gram of mouse tail tissue. We also did the experiment, which was our control, where we just coated the tail with bacteria and then took a sterile needle and just did a single shallow needle stick injury on the mouse tail through the contaminated surface. And we found actually that seemed to be a very efficient way to develop a Buruli ulcer. So 4 of those 5 needle sticks went on to develop Mycobacterium ulcerans infection. What does that look like? Here's the lesion from the mosquito bite, red. The clinical endpoint for our study was that we would stop the experiment at this point. If we let it continue, that lesion would necrose and the mouse would be in a fairly bad way, or at least its tail would. So we stop at this point. And the needle stick, again, you see that the lesion site here. So this is the summary. If you just coat with the bacteria, just the sloshing around of the tail in the bacteria, no mice developed disease. 2 out of 6 mosquito-bitten mice developed ulcers. 4 out of 5 that had the needle stick through the contaminated skin went on to develop disease. Cross-section through the tail, composite image, just showing the acid-fast staining. Here's the zone of, is the focus of bacteria. In this example here, you can see subcutaneous, quite deep. And if you look again over here, this is the needle stick injury, higher bacterial burden. These are the bacteria here growing within the subcutaneous tissue. You might also notice the epithelial hyperplasia associated with these infections, characteristic of the disease. So we repeated with Aedes aegypti, and it was somewhat disappointing because none of the mosquito-bitten mice went on to develop ulcers, even though the frequency of biting was perhaps even higher than the nodoscriptus experiments. And we don't have an explanation for that difference as yet. But 4 out of the 5 needle stick mouse, these are our positive controls, went on to develop ulcers. And we thought this was interesting, so we actually set up a formal ID50 experiment to work out, well, what was the minimum infectious dose 50 using this experimental tail model. So we took decreasing concentrations of our bacteria that we waved the tails in, and then took groups of 6 mice and did that needle stick injury. And you can see that we ended up with an ID50 of about 3 CFU. So a very low infectious dose, at least using this model. And we can just look at the odds ratios here of needle puncture versus not doing anything, and there's a very high risk, obviously, here of developing a Buruli ulcer. And if we look at the same data for our blood-fed mosquitoes, the risk is less than our needle stick, but still quite significant. And perhaps we shouldn't have been surprised by these data, because if you look back in the literature, Wayne Myers, another very distinguished Buruli researcher, recently deceased, published this very interesting article back in the 1970s from his work in the Congo, where he said that, reported a case series of Buruli ulcer patients where they'd all had antecedent trauma, and a variety of trauma, landmine injuries, vaccination, gunshot wounds, had preceded the development of Buruli ulcer. So Buruli pre-existing on the skin and then introduced into the body during these trauma events. And Wayne said in that paper, we believe that trauma is an important mode of transmitting M. ulcerans infection or introducing the etiologic agent into the dermis or subcutaneous tissue from superficially contaminated skin. And our laboratory models certainly bear that out. Okay, so Luke, what do we say to Luke? How did he get his infection? Well, you entered an endemic area, Rye, and you were probably bitten by a mosquito. And you can say, well, how was that mosquito contaminated? And we really don't know the answer to that. Was it through its development stages as a larvae? Did the mosquito then harbour the bacteria through to maturity? We don't think so. We have some lab experiments we're not going to present today to say that's probably not likely. But that it's somewhere in this, as an adult mosquito, it came in contact with fecal material or perhaps an infected possum via its lesion and then picked up the bacteria in sufficient quantities that it could spread during biting, or that Luke himself contaminated his skin and then was unfortunately bitten at that site. So this is work ongoing to try and understand these transmission pathways in a little more detail. Just in the remaining few minutes, which is only a few minutes, I should have finished, should I, Melanie? So, okay. My background is genomics, and so I just want to finish on a bit of genomics that's helped us understand how M. ulcerans are spreading in and around Melbourne. So there's sort of two possibilities to explain, the emergence of M. ulcerans. One is that it's the ubiquitous weed. M. ulcerans has been across Melbourne, all the way out to East Gippsland, everywhere in the environment. It's always there, but the environment, something's changed to make the bacteria activate. So that's the ubiquitous weed hypothesis. And, this is Patterson's curse. I'm sure I should explain the picture I've put here. Just put a picture of Patterson's curse here, a noxious weed in Victoria. And, is this M. ulcerans? It's everywhere. And then when there's enough rainfall or whatever it is, you get the weed spreading. Or is it an invading alien? Is that we've never had this condition? Patterson's curse has never been here before, and it blows in on a strong wind one day and then spreads. So we've used phylogenomics. We've gone back through M. ulcerans populations, we've sequenced their genomes and used the power of phylogenomics to understand which of these hypotheses is likely to be false. So the ubiquitous weed hypothesis, if we sequence, for example, it was present all the time. If we sequenced the genomes of these, weeds all around here and compared them, we might be able to infer a phylogeny that looked like sort of fairly equidistant radiation of the branches of the tree, because they'd all evolved, they'd been evolving at the same time. So the opportunity for genetic divergence had occurred equally amongst the population. So you'd end up with this sort of radial type tree. If we're looking at the invading alien, then there'd be a bit more structure to our phylogeny, where we'd be able to infer ancestors, and we'd have branch lengths where we would have had, if you look here at this example, in this case, the bacteria was extant in one area for a certain amount of time, developed genetic variation there before it spread to the next area and then spread to the next area. And that would reflect itself in some genetic distance in these changes in these branch lengths, reflecting this genetic diversity. So what's the situation here? We sequenced 165 M. ulcerans genomes gathered over about a 60-year time frame, going right back to the 1950s. We were able to obtain isolates from that time. And we could confirm that M. ulcerans most likely is an invading alien, or to disprove the hypothesis is not likely to be the ubiquitous weed hypothesis. And that's because this is the phylogeny we obtained. So I hope you can just see this is what we would expect for invading alien, and this is what we see for our 165 emulcerans genomes. Two broad genotypes, but lots of differences in their branch length and some structure to it as well. And in fact, from that, this is a timescale tree where we've used tip dates to calibrate a molecular clock using a relaxed molecular clock. We're able to establish when particular we could infer sort of a direction of migration. We lay onto this, so we've got time here in our phylogeny. We can also put on geography as well. And what you can see is the G2 genotype, which has the most genetic diversity. This is our Gippsland genotype, okay? It has been there the longest, had the most time to develop this diversity. And then we see these, the G1 genotype, which is the spreads in and around Melbourne. And we can unpick all that and then add some estimates of when these particular lineages arrived in certain areas. So just to summarise that very briefly, we've got arrives in East Gippsland in the 1800s. Okay, so this disease has not been in Southeast Australia for eons and eons. It's a colonial, it aligns with the arrival of European Australians. We've then got first arrival in Melbourne in the mid-1980s. Now, I talked at the start about the disease arriving in Melbourne in the mid-1980s. This aligns very well now with the bacteria arriving at that time too, invading alien. And then we have a second arrival in the mid-2000s, which was not something we knew about. So there's been two introductions from the east of the state into in and around Melbourne. We can also model the demographic expansion of the bacterial population using coalescent theory, and again, using this extended Bayesian skyline plot. This is what we really only need to look from this time point onwards, from about 2008. You can see that the bacterial population is predicted to have expanded substantially, which again is consistent with our invading alien, that the bacteria has undergone some sort of environmental amplification and expansion. And this is the epidemic curve here showing you that cases are expanding in line with our predictions of the bacterial population. So why is all this important? Why do we really want to care whether it's invading alien or, pernicious weed here all the time? Depending on which one of those things it is, the way that we approach control of this disease will change. The invading alien means if we know what the conditions are that permit it to invade, then we could design some sort of intervention. If it's here all the time, then sort of all is lost and we'll have to come up with a different control measure. So the type of agent we're dealing with will dictate the type of control measures we might instigate. So where are we now? Well, we're in the middle. We've just started a 2-year NHMRC partnership grant to really try and get a handle on what is going on. You know, you saw the epidemic curves at the start of the talk. We're on track for more than 300 cases this year. We have a group of us, research groups led out of Uni Melbourne, CSIRO and AgriBio, with the Department of Health and Human Services, Barwon Health, Mornington Peninsula Shire, to try and get a handle on all the risk factors for developing BU, whether we could conduct a cluster randomised controlled trial to intervene and clean out mosquitoes in the highly endemic areas, and if mosquitoes are an important mode of transmission, then we should disrupt disease. We're examining in more detail the conditions under which mosquitoes and other biting insects are competent vectors for Mycobacterium ulcerans. And we're trying to put all this together to develop spatial risk models to be able to help the health department predict where and when emulcerans might be likely to occur, or and stop it before it's likely to occur. If you're sitting there and you think, golly, I've got a holiday house in Rye or Blairgowrie or Point Lonsdale, should I be worried? Maybe a little bit, but the Department of Health have some recommendations, and essentially it's just to avoid mosquito bites, cover up, wash your hands and exposed limbs if you've been gardening, and if you have a suspected lesion, seek prompt treatment and suggest the diagnosis to your GP because the treatments are effective and early diagnosis is key. I'm going to stop by thanking some of the people involved here. Paul Johnson has been a key leader of the Mycobacterium ulcerans research community since the 1990s. Before him, it was John Heyman, who may or may not be in this audience, who flew the flag when no one else would. And it's not easy working on a neglected tropical disease in a non-tropical country. And actually, I think it's due to John's diligence that we actually know anything about the disease in our own backyard. So no small thing to be able to build on that. So thank you, John. Janet Fife at Vidril, Elaine Tay, epidemiologist at the Department of Health, Koen van der Lanuta at Institute of Tropical Medicine, now working with us, John Wallace, entomologist at Millersville University, Andrew Bulgens, PhD student, and Jessica Porter, RA in the lab, Torsten Seaman, long-time bioinformatics collaborator, key colleague, together with Anders, also providing bioinformatics support, and Jason Axford from Ari Hoffman's group over at Bio21. Thanks, Melanie, for the opportunity to speak here. Soft science or not, it was a pleasure. So, and I'll stop there. Thank you.
Firstly, I just want to apologise.
No, I like, that's good. No, that's all right.
Questions?
So I've got a couple of thoughts to this question and thought. So the comment is that area around the Gippsland Lakes and bands, they may also have midges that could be considered as a host, not just mosquitoes. Sandflies?
Yeah, so I think that's right. The question is, are there other biting insects that could be considered vectors? And yes, yeah. And I hope from that mouse sort of modelling that we're doing that it seems that it's the penetrating injury that's critical, perhaps not the insect per se or the.
So in the north of Australia, I think, or at least in countries north of Australia, the midges can transmit Leishmania.
Yes.
So I think and the question was, is there any report of a vertebrate host that has the mycobacterium but doesn't get the ulcer? Sort of
like, is there a vertebrate carrier that is asymptomatic? Not all possums exhibit disease. We've looked in Africa for possum equivalent. We've sampled domestic animals across parts of rural Ghana where the disease is highly endemic and not come up with anything. Colleagues have done similar things with small wild animals, rodents. Very hard to. No one's found a possum equivalent in Africa yet.
Also, for a question, do you have any? You showed your genetic data with this idea of multiple emergent populations. Do you have any data? You mentioned there's sporadic cases historically in northern Australia. Do you have any genomic data there? Is there a relationship between these? Is that an obvious potential source of the emergence?
Yeah, so the question is, have we looked in the other endemic areas in Australia, and does the genomics give you any clue as to the origin? Yes, it does. We have looked at them. Each area in Australia has its own genotype because I think the bacteria have been sufficiently isolated there to develop their own specific variations. The progression looks like evolution out of Southeast Asia into the north of Australia and then progressively south, at the same time migrating across into Africa. So it looks like Papua New Guinea has the most variation. It's not extremely well sampled, but even despite that lack of poor sampling in Papua New Guinea, we see quite a bit of genetic diversity. So it looks like it's a sort of that Southeast Asian, Papua New Guinea radiation, the time frame, wouldn't like to say, but.
So just to follow up to that, is there any relationship between, like if you said that the 2003, let's say you had one new mode, I think it was around 2003 was one of the most recent ones.
Mm-hmm.
Does that show is there any way that you can go back to the genetic data and say, that looks most like...
Oh yeah, sorry. So the 2003 introduction has come out of East Gippsland. It's a second wave.
Okay, so they're coming from East Gippsland.
They're coming from, we think, it's people putting possums in their cars and driving them to... look, I don't know, but it's something like that, or soil that's moving that's contaminated, or,
Tim, chicken and the egg. So, what originally infected the possum? So you say originates in human
Yeah.
colonisation.
Yeah, how did the possums get infected? That's Justin, yeah. No, I really can't answer that. That's a good question too, and yeah.
Any idea why it disappears from normal heads to...
Yeah, so that's another good question because the disease seems to have a shelf life in these populations. It's still at Barwon heads, but the incidence is lower than it has been. The disease was at Phillip Island in the Silverleaves area in the 1990s, and now there's no cases reported from there. So it seems that if you do nothing over time, then but that's not what we're seeing from this current epidemic. We're doing nothing or very little, and the disease is not going away. Yeah, it's moving into. Mm-hmm.
So it's more likely to perpetuate itself.
Yeah, but yeah, how are the possums? How's it getting introduced to the possums? Well, probably from other infected possums. Where are they coming from? Well, it's your grandfather sticking the possum in the boot of the car and driving it somewhere, or Justin, maybe you're a possum smuggler. I don't know, but yeah, I think that fundamental question though is how the possums got infected. We haven't satisfactorily, we haven't addressed.
I guess I'm trying to wonder where, is there a vector, something transmitting it? So is there somebody trying to infect Jason or someone trying to infect mosquitoes with it, seeing if the mosquitoes themselves? I know there's this idea that they're just
We've looked. We don't see a biological cycle in mosquitoes. We've infected first instar larvae and looked for regression through the development stages. And you lose the bacteria once they go to pupate. So there's not an active. You can see concentration of the bacteria in the midgut of the larvae, but then once they go to maturity, you lose the bacteria.
If you infect a mature mosquito.
Well, if you infect the mature mosquitoes, yeah, not really established yet what happens. Yeah.
So there's lots of possums in Melbourne. What is it about the coastal location that you think concentrates in those areas?
Yeah. No, I can't answer that. There's something though. Don't have an answer to that. We're hoping perhaps that some of the work we're doing looking at the risk factors, environmental risk factors, might help us pinpoint that. But over the journey, people have looked to try and explain that, but not come up with anything satisfactory.
The model with mosquitoes where you tip the tail, I presume you took swabs of all the pit, you wouldn't find very much of that.
Actually, that swabbing is work that's going to happen in the next few months. So, yeah. But we wouldn't expect there to be...
So we didn't expect that. Then it would have to be that the bite introduced the bacteria. So
yeah, so
why didn't you soak the mosquitoes in the Mycobacterium before exposing to the tail?
A second model that we are doing, yeah, definitely. Yeah, it's an obvious experiment. Yeah, thank you.
Hi, I'm just wondering if there's any evidence yet about the development of antibiotic resistance in these bacteria, and if that's likely given that they've downsized the genome, and if there's any work being done to understand how to wake up the immune system when you get that initial infection, and why they're not being cleared before they have killed on antibiotics.
Yeah, no reported resistance yet. There's been one description of lab-generated resistance in a mouse infection model, but using rifampicin monotherapy. But the prescribed treatment is 2 antibiotics to try and reduce that risk. But the drugs are used extensively now in the endemic areas in West Africa, and no one's reported resistance yet. There's some clinical trials going on looking at tweaking antibiotic therapy. So the microbiology's been done. I think we'd have a pretty good handle if resistance was occurring, and it's not, so that's good. But it's certainly a possibility, but it hasn't been reported yet after probably, 14 years of antibiotic treatment worldwide. The second part to the question, immunotherapy. Actually, our low infectious dose made us... People have tried various vaccine formulations, BCG being the obvious one, but then variations on that. But they've always used a challenge dose of M ulcerans of around 10 to the 5, 10 to the 6. we think that what people are being exposed to is far less than that, more like 10 to 100 bacteria. So we have just started our own vaccine program. Instead of challenging 10 to the 5, let's do a realistic challenge, and using a TLR2 agonist with recombinant protein from our mycolactone polyketide synthase, we can develop something that shows equivalent protection to BCG. So I at least think there's lots of scope there to try and maybe, do something that's immunomodulatory and perhaps protective. I don't have this crazy dream that maybe we could go and vaccinate the possums and clear out the disease that way.
So one last final comment, question?
Tim, if I can congratulate you on a magnificent presentation and say that things have come a very long way since I was last involved in the field. I congratulate you particularly on your mouse model, which I think reflects very accurately the clinical presentation with a lot of patients defining trauma of various types, scratches and so forth, not just mosquito bites. Secondly, I'd like to say that this coming and going of the infection is very characteristic in the Buruli district of Uganda now, where they had hundreds of cases every year. The disease is now virtually unknown. I also think we should mention the work of Paul Flood, who, unlike Luke's doctor, who took 3 months, 6 months to get the diagnosis from Luke himself, Paul diagnosed the first case for Phillip Island when it presented, and when that lady presented, and went on to pick more and more cases, and may I say, is still finding cases from his practice in Kew. Thank you very much for the presentation.
Oh, thanks, John. Yeah, so...
It remains for us to wrap this up, and we want to thank Tim for an absolutely amazing